Thank you very much for the introduction. So my work is going to be about sequence alignment and C-chain extend alignment and a rigorous average case analysis. So to start off, let's consider two strings of length n and m. For the rest of this talk, the larger string will be of length n. And we know that we can solve sequence alignment optimally in worst case O of m n time, right? And this can be done using dynamic programming approaches like Smith, Waterman, and Needleman-Wunsch that we've learned about. And this can be solved optimally, right, under some edit distance or affine gap costs or something like that. But if we consider something like read alignments, the sizes of our query and reference can be quite large. So for example, a human genome will have 3 billion bases around, and reads can be anywhere from 150 onwards. These optimal methods that I mentioned, they just aren't fast enough. So in practice, for read alignment, for example, we use these very good heuristic methods that run much faster. But these are heuristic methods, right? And they lack theoretical guarantees. So the theme of this talk will be, can we put non-trivial theoretical guarantees on heuristic alignment methods? And in particular, I'll be analyzing one method. I'll be analyzing the C-chain extend paradigm. So I'll be explaining C-chain extend in 3 steps. So we'll have 2 strings now, one of length s and one string of s and one string s prime. And in this case, s will be the larger string. So this diagram represents seeding and matching k-mers. On the top, we have s and s prime. As you can see here, there are some mutations or substitutions in s prime. And s prime could also be as large as s. If I match all the formers, I end up getting some diagram that looks like this. And a useful representation is representing these matches along the dynamic programming matrix. So if you look below, we have this dynamic programming matrix, and then you can have these short diagonal matches that represent k-mer matches. The red k-mer match, for example, is represented by this diagonal here. So that's the first step. In the second step, we obtain a chain, which is just a sequence of k-mer matches, and k-mer matches are also called anchors. So on the left-hand side there, this is the output of the first step, where I'm looking at the dynamic programming matrix, short diagonals of k-mer matches. And then chaining is just finding a subsequence of these k-mer matches. And on the right-hand side, I have a sequence of 3 anchors. I'll be talking a little bit more later about how to obtain this. And the third step is extension through the gaps. So in the end of my second step, I end up getting this, this nice chain, but it's not a full alignment yet. I need to complete it to a full alignment. And how I do that is I do dynamic programming through the parts that are unmatched. You can see these green matrices here, and those are the dynamic programming matrices that I go through in order to get my final match. And once I do dynamic programming on the gaps, I get something like this. This is the full alignment, and it describes the entire alignment of the sequences I care about. So this paradigm is super useful. It's used in read-to-genome mapping, such as Minimap2, and it's also present in genome-to-genome alignment, such as mummer. And although these algorithms differ in exactly how they go about alignment, the core idea is very, very similar. It's this kind of C-chain extend paradigm. But the worst-case behavior is still quadratic. In reality, if you run any of these programs, it's much faster than this, right? So this worst-case runtime doesn't actually explain the better than worst case runtime that we see in practice. And to get through this gap in reality and worst case analysis, we turn to average case analysis instead, where we ask, how is the runtime on average and not just in the worst case? But to talk about average cases, I'll need some sort of random model to take expectations over. And so now S is my reference genome. It's a uniformly random string of nucleotides of length n. So I just pick nucleotides uniformly at random, and I'll have a read S prime, which is a substring of S of length m. I sample a substring, and then I introduce substitutions independently at random along this substring. And finally, I'm going to let the k-mer length scale with n, the reference genome. It's going to be something like log 4 n times c, where c is a constant that depends on theta we describe in our paper. You can think of it as approximately being 2. So to analyze runtime now, we have to analyze the sum of these 3 steps that I've described. But the big caveat is that these runtimes, they're not deterministic anymore under our model. They're random variables, right? These runtimes depend on what random strings get spit out by my random model. So I want the expected value of this sum. And in this talk, I'll be focusing on the time of chaining and extension, because in the paper, we justify that both theoretically and practically, the time of seeding is relatively small compared to the other two. So there's two questions that I want to answer first off, which is what is the expected time of extension, and how good is the resulting alignment? Because if the resulting alignment isn't any good, then it doesn't matter how fast it is. And to do this, I'll go back to this dynamic programming matrix formulation. So I have my string S on top here, and then I sample a substring that looks like this, and the bolded letters are mutated, right? So I have my matrix here, and I want to highlight this diagonal. I call this diagonal the true homology between the strings, because I'm sampling a string, right? I'm sampling a read from a reference, and I introduce mutations. I should really be aligning back to where I sampled from. That's where the homology really is. So the goal is to hopefully align to this diagonal. So if we look over, standard programs, dynamic programming, in this case it's a semi-global dynamic programming. The point of dynamic programming is that it's good because I search the entire space of possible alignments, and that's why I get the optimal one. But that's also a drawback because that means it's slow. I have to search this entire matrix that it's got size O of m times n. A heuristic people often use is banded dynamic programming. So instead of searching the whole matrix, you just search a band instead. And this is good because it's fast, right? It scales with b, the band length. But if you do it naively, and I'm trying to do read alignments, I don't hit this diagonal, right? I want to align through this diagonal. So that's not good. I'm missing what I want to align to. And what C-chain extend does, you can think of it as combining the best of both worlds. So for example, I have my string here, and I have my substring sampled over here. And the blue bolded bases represent k-mers. They're formers that match. And over here, I get a nice chain that lies in this diagonal. And the extension time is relatively short because I have these small gaps I have to fill in, but these gaps are small. I don't have to fill in the entire matrix. So that's why it's nice and fast. But in the worst case, things can still go wrong with C-chain extend, right? In this case, I end up getting my string here, and I got a huge gap. I only have two k-mers, and they're off diagonal. So bad things could potentially happen. And in this case, the runtime will be something like O of m times n. But the point is that we care about the expected size of the search space. So even if it's bad sometimes, if it's bad rarely, that's still OK. And secondly, I want to talk about recoverability. This is a new metric we define, which is the fraction of the diagonal covered by the search space in the k-mers. Intuitively, what this means is, can our alignment hit the diagonal that I care about that comes from my mutation model? Right. So theorem 1 from our paper is our main theorem, and it's that the expected time of extension is this term over here. It's m times n to the power of f of theta log n. F of theta is some function that depends on theta. We give it in closed form in the paper, but a nice upper bound is that it's less than this multiple of theta. And what you immediately see is that when theta is small, the mutation rate is small. This is strongly subquadratic. The expected recoverability is this term over here, and this basically means that as my read gets longer, I'm guaranteed to have a nice, it's going to be recoverable, and it's going to be mostly on the diagonal. So the proof idea here, I won't go too deeply into this, but the idea is that we have sums of k-mer random variables that give rise to these matches. And k-mers are not independent, as you've heard about in the previous talk, because one mutation affects multiple k-mers. But they're almost independent in some sense. And we can bound almost independent sums of random variables with high probability using some more sophisticated mathematical tools in these papers. I'll give a link to the references at the end. And then we also use some tools from Ganesh and Sai to do some combinatorics. So once we bound a way to degenerate chainings with high probability, we want the expected time of extension. And this is essentially the square root of the gap size, right? Because the matrix is the square root of the gap size. And this turns out to have this nice form. This 1 minus theta to the power of negative k is quite interpretable. It shows up when you talk about k-mers and random mutations. And then if you remember that k is c log n, and you rearrange the formula, that's how you get this final term over here with the f of theta. So I need to finish off by talking about the time of chaining. So we'll let capital N be the number of k-mer matches or anchors, same thing. And remember that capital N is a random variable, right? The number of matches depends on what string pops out. If I have a string of just only A's, then I'll probably get more k-mer matches. And we're also using the linear gap cost objective here. We describe more about the specific model assumptions we're making, but we're using a linear gap cost chaining objective, which is really the simplest, non-trivial chaining cost there is. And the nice thing about this is that this can be optimally solved. So I'm finding the optimal chain in capital N log capital N time with some nice data structures. But under this chaining objective, theorem 6 in our paper says that the expected time of chaining is also small. It's also much less than quadratic. It's m log m. In fact, this is a loose bound that I put up here just because it's nicer. We actually have a slightly lower bound, a better bound in our paper. But same story, it's fast. Okay, so we have these asymptotic runtimes in an expectation, and the question is, can we actually do anything with this? So what we did was we took real nanopore reads, not simulated, real nanopore reads of about 95% identity, and we built a custom C-chain extend aligner with k being this value, as I mentioned before, and we aligned some reads. So we aligned reads to a few different species, and on the top row is the extension times, on the bottom row is the chaining times, and the x-axis is the read length. So I'm looking at how the read length affects the runtimes. And one thing I want to draw your attention to is this bottom right corner over here. So that's chaining time for humans. And as you can see, it's a lot larger than the other species. And that's because our random string model starts to break down when it comes to humans. Humans have a lot of repetitive elements, and you can kind of see that over there. But what I want to talk about is this chaining time. I want to talk about this. So the expected time of chaining under a model is this term over here. And f of 0.05 is about 0.08, and we show that in our paper. The slope of the extension time should be proportional to that red term over there, right? Because I'm plotting on the x-axis the read length, so that slope should be that red term, approximately. Is this true? So what we did was we plotted all the slopes of those extension times in these, we have 5 data points, and we fit 2 different functions. We fit a naive function, which is just a log with 2 parameters, and we fit the log term with the 0.08 correction that's predicted by our theory. And what you see is that this predicted curve is actually curved in log space, and it fits our data better. And the cool thing here is that we didn't try different values for 0.08. This just popped out of our theory, and it fits better. And furthermore, nanopore reads have indels. They have a lot of indels. We didn't model indels at all, but it seems like indels are somewhat accounted for by our theory. So this means under even modest model misspecification, it still works, which is nice. The last thing I want to talk about is sketching. So we talked about sketching earlier, which is fantastic. I don't have to go too much into it. But real long-read aligners, they don't actually use all k-mers. They subsample the k-mers with sketching, and they use these techniques like minimizers and syncmers that I won't get into. But this is what sketching looks like graphically. You just take a subset of the k-mers. They're overlapping. You don't need all of them. So we just take a subset. Our theorem on sketching says this. So we let C scale with log n, but it's less than k. And we sample to 1 over C of the k-mers. So out of every C k-mers, we sample about one of them. So under this assumption, with the open syncmer method, which I won't describe, but is a method of sketching, we get the exact same runtime and recoverability results, asymptotically. They don't change at all, but the chaining time actually becomes provably faster, which makes sense. If I take 1 out of 15 k-mers, chaining is 15 times faster. So sketching reduces chaining time without increasing extension time too much. That's essentially what this theorem states. So how reasonable is this? Because all of our results are asymptotic. Can we actually get some non-asymptotic results? And the answer seems to be yes. So what I did here was we simulated reads of length n on the bottom, or genomes of length n, and then we had some mutations, and then we just aligned 2 length n genomes. I'm sketching with increasing density. So I'm sketching with about 2 log n density. And what this means is that as my strings increase, I'm sketching more and more. And this red line is the chaining speedup. So in this part over here, I'm about sketching 1 out of 18 k-mers. But I'm only increasing the extension runtime by about 3 times, and I'm decreasing the chaining runtime by 18 times. So that's a really favorable trade-off. I can decrease the chaining time a lot without increasing the extension time too much. So I want to conclude now. So what we did was, so our motivation was that optimal sequence alignment is slow, and in practice, no one just uses Smith-Waterman on reads against a reference. But we use heuristics instead that have bad worst-case guarantees, and they're not necessarily representative of real times. Or in real read aligners, they just throw out the cases where you end up getting really bad runtimes. So what we did was we proved a strongly subquadratic runtime in expectation which allows us to get around the worst case bounds. And we have good accuracy under our recoverability metric, and that our bounds also work for sketching and real data and provide some justification of sketching and long read-aligners. So lastly, our paper is, this is the title of our paper now. It's published in Genome Research in Advance. Thank you to the anonymous reviewers for encouraging us to work on real genomes more. This is me. This is my advisor, William Yu. Thank NSERC and University of Toronto for funding. This is a QR code for the posters and the references. So, sorry, the slides and the references. So if you want to take a look, feel free to do so. Thank you very much.